Okay, so let's briefly talk about nutrient requirements. I'm not going to really go into a major chemistry review, but these 6 atoms are the most common elements. In addition, you need some trace elements such as magnesium and calcium, things like that. But carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur make up the bulk of the cell. And so, again, you know, a lot of these carbon-carbon backbones make our lipids, sugars, and proteins. There are some inorganic molecules we need and some ions. We can actually sort of classify and distinguish species by where they get their chemicals from. What are nutrients for? Obviously, we need them to synthesize new cell components, so we need new membranes, cell walls, proteins, nucleic acids. And we get that by breaking stuff down into the component subunits, so sugars, amino acids, and nucleotides. But what is interesting is that prokaryotes, so primarily bacteria is what we'll discuss, but they exhibit this huge diversity of metabolic abilities. So some of them are really different in how they get their energy, where they get their materials, what things they can make from scratch, what they need to be given in the food if you're going to grow them, and what's available in their normal environment. So every living thing on Earth needs carbon, obviously. It's about half the dry weight of the cell, and it's in absolutely everything. The carbon-carbon bonds store the bulk of the energy, but different groups of prokaryotes are distinguished by where they get their carbon. So heterotrophs use organic carbon sources. What does that mean? What is an organic carbon source? Right, so there's carbon attached to at least one hydrogen, and usually there are carbon-carbon bonds as well. So their organic molecules are at least methane, at the very least, when there's just a carbon with 4 hydrogens, but usually much more complicated carbon-carbon molecules. Autotrophs, on the other hand, get their carbon from carbon dioxide in a process called carbon fixation. Which category are we? Which way do we do it? Where do we get our carbon? We are heterotrophs. Animals are all heterotrophs, fungi are heterotrophs. What are autotrophs that you know of?
What's that like saying? CO2, I don't know what they're called.
Sure, and all plants, right? All plants are autotrophs. Yeah, they use carbon dioxide and they make glucose out of it, right? They sort of are the basis of our entire ecosystem. Now, think about that. If you're getting carbon from organic carbon sources, which are high energy molecules, versus getting carbon from carbon dioxide, and again, why are we getting the carbon? What are we going to use it for? We're going to build stuff, okay? Now, if we want to build stuff, and it's partly already built, like if you're building a polysaccharide from glucose, that's way different than building a polysaccharide from carbon dioxide. Which one is going to cost more? Which one's going to take more energy, use up more ATP? If we're building from carbon dioxide or building from, I don't know, ethanol. Yeah, we're going to need a lot more energy to start with 1 carbon at a time. And so those organisms, those autotrophs, are gathering and they need a lot more energy. So where different organisms can also be classified based on where they get their energy. And this is tricky to understand because for humans, the source of carbon and the source of energy are the same thing. But that's not always the case. So what do I mean by that? What do I mean when I say that in humans and other heterotrophs, the source of energy and the source of carbon is the same?
That's how we get our energy also from the
Yes. So we get energy by chopping up carbon-based molecules, and we also use the carbon in the carbon-based molecule to build stuff. But if we are an autotroph, we get our carbon from carbon dioxide, and we get our energy from sunlight, for instance, or from some other place, from some chemical compound. So phototrophs obtain energy from sunlight. This term phototroph is referring to the origin of the energy. Autotroph refers to the origin of the carbon, right? So you can have an autophototroph, but you could also have a heterophototroph, for instance. So they get their energy from sunlight. That includes plants, single-celled algae, and photosynthetic bacteria. So there are some bacteria that do this. Chemotrophs extract their energy from chemicals, so from breaking bonds in chemicals. So that would include us and the cells that we're made of, fungi, and many types of prokaryotes do this. The kinds of chemicals we use include sugars, amino acids, fatty acids. Some prokaryotes use inorganic chemicals like hydrogen sulfide and hydrogen gas as a means of gathering their energy. So phototrophs are ones that extract energy from sunlight, and chemotrophs extract it from chemicals. So I just want to make sure that you understand the difference between energy source and carbon source. People get them frequently confused. So if we add up these things, we can create sort of a variety of different classifications. So for instance, a photoautotroph, what is that telling us? The phototroph part is about energy. So where are they getting their energy? From sunlight. An autotroph is referring to where they get their carbon. So where are they getting their carbon? CO2, right? So these are guys that use CO2 to make carbon-based molecules and get the energy to do that from sunlight, such as what? Plants, anything that does photosynthesis. Photoheterotrophs. They get their energy from where? Sunlight. They get their carbon from organic compounds, so they don't use carbon dioxide. There are some bacteria that do this, that use the energy from sunlight, but they get their carbon instead of using carbon dioxide. Chemoautotrophs are also in the bacterial world. Chemoautotrophs means that where are they getting their carbon? Carbon dioxide. Where are they getting their energy? Chemicals, okay? So they get their energy basically from rocks, and that's why they're called rock eaters as a sort of slang term. So they use the energy in inorganic compounds, and then they use that energy to make stuff straight out of carbon dioxide. So this is about as far as you can get away from human metabolism as is possible. Chemoheterotrophs, they get their energy from chemicals. They get their carbon from organic compounds. So they're coming from the same place, basically. And that includes humans. To remind you what nitrogen is important for, it's found in amino acids. Which part of the amino acid includes the nitrogen? The amino group. Most bacteria can break proteins down and get nitrogen that way. So this again, how do you get your nitrogen? Most of the bacteria do it the same way we do it, which is by breaking down proteins. But some bacteria use ammonia or nitrate. Do we do this? No, not really, not typically. We can make those as waste products that we then have to deal with, because often we'll metabolize amino acids by lopping off the amino group, and then now we have ammonia. And ammonia is terrible, we have to get rid of it, so we turn it into urea and excrete it in the urine. There are some bacteria that use nitrogen straight from atmospheric nitrogen gas, and that is known as nitrogen fixation. So whenever we're talking about carbon fixation or nitrogen fixation, we're talking about taking it the simplest form that's readily available and turning it back into something that other organisms can use. Sulfur is also important. It's found in a couple of amino acids and in some vitamins. Most bacteria get sulfur by decomposing proteins because, again, it's in some amino acids. So if you break down cysteine, for instance, or methionine, you can get the sulfur out. Some bacteria use sulfate or hydrogen sulfide gas, which we cannot do. Also, phosphorus is important. Where is phosphorus found? It's found in nucleic acids, DNA, and RNA, and what else? Phospholipids, and what else? It's also super important for energy. So usually phosphate is just an ion that's floating around in nature, and that's where we get our phosphorus. So if we fed radioactive sulfur to bacterial cells, where would we find this sulfur when we did a chemical analysis of the cell? We would find it in the proteins. We also need some trace elements to make some enzyme cofactors, specifically these trace metals. And growth factors. Growth factors are anything that you cannot make yourself, that you just need to have provided in the media. And a lot of microbes can't make certain things, but those things can be different depending on the species. So there's certain amino acids that they can't make from scratch, certain vitamins, certain components of the nucleic acids that they may not be able to make and have to just be in the food. And those, by definition, just means they're things that if they're not provided, then the organism won't grow. Now, microbes tend to be very diverse if you compare different species to each other. So here's an example of E. coli, which is capable of making all of the things that it needs from glucose, basically. Obviously, it's going to get nitrogen, sulfur, and phosphorus, a couple of other places, but it can make pretty much almost everything from glucose, and it's very broad metabolic abilities. Neisseria, the genus that includes the gonorrhea bacterium, is extremely finicky, and you have to give it almost everything. So that means that its environments can be very restricted. In fact, it just so happens that the restrictive environment, say, of Neisseria gonorrhea is the nice cozy place inside your body that you would rather it wasn't growing. So these guys are termed fastidious. They're very complicated and hard to culture. What is the basis of these differences? If we look at a chemical level, what's different between E. coli and Neisseria gonorrhea? Yeah, so which metabolic pathways you have? Which means what? You have either like these 6 enzymes, and these is only the things you can do, or you have all of these enzymes, which means you have the genes for making only these things, or the genes for making all of these things, right? And we're going to see that difference manifested in the proteins. All right, so let's move on to culturing microbes in the lab, and then we can measure growth. We need a medium. We're going to culture today from several different kinds. We need it to be sterile to start with, obviously. We usually autoclave it. If any of you have worked in a medical facility, you've probably seen this happening. The inoculum is just the thing that you take from, usually from a pure culture, but it could be inoculating from the environment. And that's whatever it is that you want to grow, that inoculum is then put onto the media that's initially sterile. And then we culture them. So here are the different types of culture media. Complex media are rich. They have a lot of stuff. Their exact composition is actually variable from batch to batch because it's not like you have a bunch of pure ingredients like you have sucrose and you have, nitrate and then you have sodium chloride and then you measure it all out and you put it together. Rich media is typically made from taking, say, milk and partially digesting it and throwing that in, and maybe you take a little beef broth and you throw that in, and you add some other stuff. Okay, so it's variable, but it's rich. Lots of things can grow on it. As opposed to a chemically defined media where, again, you measure out exact amounts of pure chemicals so you know exactly what's in there. And so this would be useful for controlled experiments. This is going to be useful for just growing stuff, a lot of different kinds of things. If you had to culture something that you hadn't worked with before, which of these would you probably use? The complex, right? Because you don't know at the outset what its cultural requirements are going to be, so you have to just give it as many different things as you can. We grow them in this lab a lot of times on solid agar. And you'll see we have dishes, slants, deeps. You'll work with all of those. All right, so there are a ton of different media. We can make lots and lots. Here are just a very small sample of the different kinds of media. I like that one, always makes me hungry. I don't actually eat it, so don't get any ideas. But despite this ability to make lots of media, there are tons of prokaryotes that have yet to be cultured in the lab. In fact, the vast majority of single-celled organisms out there cannot be cultured yet. So we're going to talk about special types of media that are useful for either identifying or isolating species that we're interested in. We need to talk about the difference between differential media and selective media. Differential media is something that basically has substances in it that allow the microbes that you're growing to be identified. Usually it means that you're going to grow most everything. But some things are going to have a different metabolism than other things. And then you're going to see some visible difference either in the colony color or the color of the surrounding media that's going to tell you it belongs to this category or that category. And so in that way, it's a lot like a differential stain, right? We're sort of classifying organisms. Now, selective media, the easiest way to describe what this does is that the outcome isn't, red and green, like different colors or different physical features that you can tell things apart. This is going to kill some things and allow others to grow. So it's a dead or alive test. Yeah.
Is it the solid one or the rock, or could be both?
Could be both. We'll talk about, so today we're going to talk only about solid examples, and that's the most common. But you certainly could imagine a broth that if the organism can do this, the broth turns that color. If it can't, the broth turns a different color. So differential media allow us to distinguish between different organisms. Generally speaking, both of them are growing, but depending on some visible difference, either the color of the colony, the color of the agar, whether there's a clearing around the growth, some visible difference will allow you to tell the difference between them. Does that make sense? So that's what it is. It's not just a color change, although it is the most common thing that we'll see, but there can be other things that would qualify as differential media. Some media are both selective and differential. An MSA that we'll talk about today, mannitol salt agar, has 7.5% salt. So remember, what is the natural percent solution inside the cell? Usually 1. So if you have 7.5% salt outside, is that a normal, high? For most organisms, it's going to be lethal. Now, we did talk about halotolerant and obligate halophiles and extreme halophiles, which would be those organisms that can tolerate or even prefer high salt. So there's the salt in there that will allow us to tell if something can survive high salt. That is going to be the selective part, right? Because if you can't survive high salt, what happens to you? You die. Okay, so dead or alive, that's the selective part. This is a little harder to explain. So this media also contains mannitol. You may not know what that is. I'm just going to tell you it is a sugar. And like all sugars, some organisms can ferment it. And the fermentation process, which is varied among lots of species, is one way by which we can identify different organisms. So there's mannitol and a pH indicator. If we're asking the question, can you ferment mannitol? Some organisms can and some can't. Now, regardless of whether they can or can't, they're going to be able to survive. But if they do fermentation, what happens when they metabolize mannitol and do fermentation? What is the usual outcome of fermentation? Say again? More acidic. So the product that you make by taking, mannitol gets turned into something that can be part of the glycolysis pathway and ends up as pyruvate. We have to regenerate our NAD. So we put our electrons on pyruvate with some enzyme, which in this case makes whatever acid it makes. And if the medium becomes more acid, we can tell that because there's also a pH indicator. The pH indicator is just a thing that changes color at different pH. If you've ever used a hot tub, you know about pH indicators, right? If you have the water and you're trying to decide if it needs more acid or more base or whatever, you just put the pH indicating strip into the water and then it changes a certain color depending on the pH. So there's a pH indicator that is red at neutral pH and yellow at acid pH. So an organism that can grow on MSA, like Staphylococcus epidermidis, which is halotolerant, so it's going to survive, and its friend, Staphylococcus aureus, which is also halotolerant, this guy turns the media yellow, this guy leaves it red, so which one of them can ferment mannitol? Yeah, if the media turns yellow, that's because this guy is taking the mannitol, fermenting it, producing acid. The acid gets secreted into the medium, it diffuses out, and the pH indicator changes color. All right, some other things we need to think about are temperature and atmosphere. I'm going to very briefly go through this. So most organisms that live in room air prefer about 20% oxygen, which is what we like. So when we grow them, we shake the cultures to get oxygen in there. There are many medically important bacteria, such as Neisseria, which includes that gonorrhea pathogen, and Haemophilus, which causes a bacterial type of pneumonia, that grow best with a little bit more CO2 than is in room air. So if we're going to culture them, we actually will provide them with more carbon dioxide. These guys are called capnophiles. That just means that they like carbon dioxide. And one way you can grow them is just in a very simple candle jar where you're burning something that's producing a little carbon dioxide. When you want to culture microaerophiles, these guys require less oxygen than is in room air, and that you can achieve using this simple apparatus. So there are a variety of other ways to make sure the oxygen concentration is right. Okay, last we're going to talk about enrichment media. That is, if we have a mixed culture, say we're taking a sample from the environment or from someone's, sputum or whatever, and the thing that we're interested in culturing is in really low abundance, but still it's enough to make you sick. You can actually enrich for that rare guy by providing him with something that he's especially good at using that other organisms can't use. Now, one example of that is blood. A lot of pathogens, not a lot of them, but some of the human pathogens are really good at getting food out of blood cells. What they do is basically they secrete enzymes that explode the blood cells, and all the good nutrients inside the blood cells leak out and they can live on that. But most organisms can't do that. So if we wanted to enrich for those guys, because we suspect someone had a streptococcus infection, for instance, then we could provide blood in the media, and the guys who can use it, our suspected pathogens, are going to grow faster than the other ones, and eventually we're going to be able to culture them and prove that they're there. All right, couple of definitions. We need to know what a pure culture is. So what is that? What is a pure culture? It's one species, and by definition, actually, when we do it in the lab, we are actually culturing the entire thing clonally from a single original cell. Okay, so we allow one cell to be on the plate. It grows into a colony. They're all derived from clonal divisions of that one cell, and then we take that colony and we culture that. Okay, so and we just keep going. Now, as longer time periods elapse, those individual cells can evolve away from each other. Right? You can introduce little mistakes in the DNA replication process, but over shorter time spans, we can maintain pure cultures that way. What is a colony? What was that colony term? What does that mean? That was that term from the plate, right? So that one cell is dropped on a plate, and then it grows by binary division, right? By binary fission, just clonally into a little cluster of about 1 million cells that are all related to each other. Now we're going to use this method, the T-streak, also called the streak plate method in your lab manual, to isolate pure cultures. We're going to do that today. I'm going to go into this in more detail when we talk about the lab, but this is sort of just an overview of how you do it technically and what it ends up looking like. So there's lots of cells here, and then we're diluting. We actually sterilize our instrument at this point. We go grab a little bit of culture from here, we dilute it some more, sterilize again, grab a few cells from here, dilute, and then at the end we end up with just individual cells on the plate. Okay. Last question, how do we measure growth? So there are a number of measures of growth. I'm going to talk about them, and also they're covered in some detail in the supplemental module that's provided for this chapter online. And so you can also do the problems there to prepare you for any questions that were sort of math or logic based that you might see on the test. So there's a couple of ways to look at growth. Direct ways and indirect ways. What do you think the difference is when we use this word direct or indirect? What is that referring to? What would be like sort of a definition of direct methods of observation? This is basically when you're actually eyeballing the growth. Whether you're eyeballing the individual cells or the individual colonies, you're actually looking at the growth yourself, right, and counting it in some way. Now, indirect methods are estimates based on some property that you can measure, okay? Maybe light scattering, through the culture, or maybe how much chemical activity they're doing, or how much they weigh. So the simplest, and this was one of the first things I ever did in the lab actually, and it is really annoying, but it is fairly accurate. The simplest is doing direct microscopic count. So this is where you're actually laying eyeballs on the cells themselves using a microscope. You have this little slide, and it's got a certain volume, and each square is a certain volume of the culture. And then it's divided in a grid so that you can easily count. It's fairly easy to just count these. And then you'll get a certain number, and you know the volume of this chamber, right? So then you can get a number per volume. And that gives you the concentration directly by counting it yourself. Another way to do this is by looking at colonies instead of individual cells, but we can still count cells because these colonies each came from one cell. So by counting colonies, we're still counting cells. So again, we have our starting culture, and we take some volume of it, and we put that volume onto a plate, and then we look to see how many colonies there are, which tells us how many cells there were in that original volume. Now, the downside of that is that if this original culture was really, really dense, then when you take something like a mil of it and put it on a plate, then how many cells are you going to be putting down? Potentially like millions. Okay, so is that going to be possible to count? It's not even going to be possible to see, because when you put a couple 100,000 cells on a plate, they just grow together and form a lawn. So what you have to do then is you have to take this original culture, and since you don't know at the outset what the actual concentration is, you have to do serial dilutions. So you take your culture and you do a 1 in 10 dilution. So maybe you take 1 mil into 9 mils, and then there's 10 times less in concentration in this new tube, the 1 in 10 dilution. Then you do another 1 in 10, so now it's 100 times less, and another 1 in 10, so 1,000 times less. And you make a whole bunch of those, and then you take known volumes from there and put them on the plate. And then you look for the one that you can count. And then you can extrapolate back to what the original concentration was. Does that make sense? I'm going to show you what that actually looks like. So here's our original inoculum. It's taken into this tube. 1 mil into a total of 10 mil volume is a 1 in 10 dilution. Then we just do serial dilutions, diluting it down further and further. So the most dilute one is 1 to 100,000. Then we take a milliliter of this dilution, and that one, and that one, and that one, and we put it on a plate. Now what's happening here and here is that we have so many cells still in this dilution that it's all growing together in a line. But at a certain point, there's enough dilution that there's a countable number like 30. Okay, so then we can say, oh, there's like 35 on this plate. It's a 1 in 10,000 dilution, so I know that it's like 350,000 in the original concentration. And you would have to know how to do this math. It's very simple. Now, a couple things to note. You might be tempted to count this one, but you're not going to be accurate because there's regions that you can't count. You might be tempted to count this one. Why would this be bad to do? Because statistically, when you get into those low numbers less than 30, the variation is going to really skew your results. Because the same culture could just by accident give you 1 colony or 7. And that's going to cause almost a whole order of magnitude difference in the number that you calculate. Whereas if you're counting, a couple extra cells here, it's still going to give you roughly the same number at the end. So for instance, this plate here, the 1 in 10,000 dilution gave you 34 colonies, which is a perfect number. And we just take 34, we multiply by the dilution, and we know what the volume was, and that is our count in the original culture. You can also count by membrane filtration, where you take a known volume, you filter it, and then you row the filter, and you count. And this is going to be something you would do if your colony-forming unit was really dilute, so that you can concentrate it. And then you know that, say you filtered 5 liters through this filter, and then you got, 75 colonies in 5 liters, and you can do the math to figure out what the original concentration was. All right, MPN's most probable number is a statistical way of doing this kind of, it's similar in principle to the serial dilution colony counts, except that it's in liquid culture. And what you do is you do a bunch of serial dilutions, but in replicates of about 5. And you keep diluting until your replicates start to not grow anything, which means in these tubes, how many cells were there? How many cells in these tubes that didn't grow anything? None, okay? And in this more concentrated, 10 times more concentrated dilution, there's a couple of tubes that had 0 cells. So what you're not really counting directly the number of cells, you're counting how many tubes had no cells in them versus the ones that had at least one. And then just by doing statistics, you can figure out how many likely cells you had to start with. So that's why it's called probable rather than actual. And you would just use a table. And there's a problem set includes a fairly detailed worked problem of this kind. So you might be required to just be able to use this table. It's fairly straightforward. Okay, now the indirect methods. Turbidity is the most common indirect method, and that is basically, here's your culture. This is your negative control, right? This is just broth. And here's your culture with all cells in it. And if you shine a light through the culture, which one is going to get more light passing through? The one without any cells in it, because the cells are in the way, they're blocking the light. And if you just measure how much light you get on the other side here versus there, then you get an absorbance. And by doing some controls and some calculations, you can use this optical density to figure out, actually with very high degree of accuracy, how many cells you started with. So we can also measure biomass. If we know kind of what's in a cell, once the water's been taken away, then we can also figure out, like, if you have 6 kilograms of dry weight, then how many cells was that to begin with? We can also do things like, if we know that cells under optimum conditions always produce this much carbon dioxide or this much acid, we can measure how much there is and figure out that way how many cells there were to start with. Okay, last point is that we have to sort of come back around. We've been talking a lot about how to culture in the lab. So why is the lab not a natural environment? What's different between growth in the lab versus outside in the field? Yeah, there's fluctuations in everything. Temperature, degree of water, even how much oxygen there is. If you get stepped on, right? There's a huge amount of variables that are not controlled. Nonetheless, it's really important to understand what happens in nature because that's where most of the microbes are found that do stuff importantly for the ecosystem. And there are things that happen in nature that don't happen in the lab that are very hard to simulate in the lab because most microbes grow in a community. They're not pure cultures. So cells can respond dynamically to their environment, and if you don't provide a dynamic environment in the lab, you can't really learn anything about that. And they also typically live in community, and we often refer to those as biofilms. And the biofilm is a mixed community that has a lot of polysaccharide coating around it. It's the glycocalyx, basically. So examples of biofilm would be that slippery gunk on the rocks of the stream that when you're hiking wants to kill you. The stuff that clogs your sink drain, the scum that builds up in your toilet bowl, and that stuff in your mouth that you brush off every day. All of these are natural, normal biofilms that usually have multiple species in them. And the reason that they're slippery is because they have a lot of sugar coating, and that's attracting a lot of water. And when there's multiple organisms living in community, they tend to sort of collaborate, and they are sort of stronger as a whole community than as an individual species. So when we look at biofilms, there's a few things we'll notice, right? We'll notice that there are actually channels for water to get through. That makes sense. The cells are talking to each other using chemical signals. And this is important. Obviously, biofilms have importance to us medically, and a lot of infections in your body are actually biofilms. They're not just one sort of pure culture. In particular, ear infections are typically biofilms. All of the stuff that accumulates in plumbing is important industrially. And biofilms are important from the microbes' perspective because they're helpful in protecting them. In fact, if you're in a biofilm as a microbe, you are typically much more resistant to chemicals that we're trying to kill you with. So that like scum in your toilet, all of the microbes in there are are more protected from the things we're trying to use to get them off than they would be if they were just a pure culture coated on the inside. Biofilms are also important if we're trying to sort of simulate natural things that microbes can do that are useful. We have to be cognizant of what conditions they prefer, and usually they like to live with other creatures. So those interactions between the members of a microbial community can be cooperative. So for instance, if you're a strict anaerobe, you can still be growing in the mouth of a human if the other members of the biofilm are taking all the oxygen away and sort of making it safe for you. Some organisms' metabolic waste are food for others, but the interactions can also be competitive. So in many cases, organisms secrete toxic compounds to keep their competitors away so that they can hog all of the nutrients in their environment.